The Galien Foundation fosters, recognizes, and rewards excellence in scientific innovation to improve the human condition.
André Malraux said once in one of his novels, "Une vie humaine ne vaut rien, mais rien ne vaut une vie humaine." Human life is worth nothing, but nothing is worth a human life. All these men and women are trying to save human lives. Is there anything more noble than that?
The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year Best Pharmaceutical Product, Best Biotechnology Product, Best Product for Orphan Rare Diseases, Best Medical Technology, Best Digital Health Solution, Best Incubator Accelerator Equity, Best Startup. Around the globe, the Prix Galien is considered as the equivalent of the Nobel Prize for the industry, mobilizing an unrivaled network of Nobel laureates and top biomedical scientists. The Galien Foundation manages an independent, cross-functional, and geographically diverse program of events and sponsorship to brand the best of the best in new medicines and diagnostics. The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Roy Vagelos Pro Bono Humanum Award for Global Health Equity is bestowed to an individual, a company, an academic institution, or a non-governmental agency that has helped to improve the human condition through the application of biopharmaceutical science to problems of developing or underserved populations worldwide.
This is the right event, on the right issue, at the right time.
Staging this event in Africa is a significant statement that science and innovation are needed as much here in Africa as anywhere else.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Good afternoon, everybody. May I welcome you to this Galien Foundation webinar, where we will be focusing on the challenges of CNS drug discovery, which often take longer to develop and have a lower success rate than other areas of drug discovery. What are the strengths of the nominees that are working to win this award this year? How have they been able to de-risk the CNS therapies they're working on? And how have they been able to speed the research and development pathway through the clinic to market? Are areas that we hope over the next wee while to learn more about. My name is Hugh Marston. I have the pleasure to be the Global Head of CNS Discovery Research at Boehringer Ingelheim, and I will be moderating this afternoon for us all in this very exciting webinar. I have two excellent speakers who have both got products that are looking to win in this category. And if I would like to just pass over and introduce the two speakers. First, I'll go to Michelle from Amylyx. Michelle, can you tell us a little bit about yourself and the product that you're proposing for?
Sure, Hugh. Thank you so much for that introduction. First, I'll thank everyone for your patience, as my voice quality is not very good today. I am the Vice President and Global Head of Medical Affairs at Amylyx Pharmaceuticals. It's a pleasure to be here. Amylyx is honored to be considered and recognized by the Galien Foundation, and thank them for this opportunity. We also want to express our gratitude for the partnership and continued support within the ALS community and past and current clinical trial participants without whom our work would not be possible. I think for Amylyx, our story begins in 2013 when our co-CEOs, Josh Cohen and Justin Klee, founded Amylyx. And they really did this all based on a research question. And that is, what makes neurons die? And clearly, this is a central challenge in all neurodegenerative diseases. And at the time, there was a great deal of effort being put on causation, for example, genetic studies, and that is still ongoing. But there wasn't a lot of attention paid to slowing the progression of neurodegenerative diseases, which is a key hallmark of these diseases over time. Their hypothesis became, what if you could address misfolded proteins, protein accumulation, and mitochondrial stress that often leads to apoptosis and neuron death? Would disease progression be slowed if you could halt this neuron loss based on these mechanisms? So they poured over an endless library of scientific research and medical journals looking at what had been done in the past. And they actually identified two existing molecules, sodium phenylbutyrate and taurursodiol, also known as TUDCA. And the scientific rationale for these just became stronger and stronger as they continued to investigate. Sodium phenylbutyrate stabilizes the endoplasmic reticulum protein folding and reduces ER stress and modulates the unfolded protein response, while taurursodiol incorporates into and stabilizes the mitochondrial membrane, which increases the apoptotic threshold of the neuron. They were able to undertake some preclinical work for proof of concept to show the effect on preserving neuron viability in preclinical models, and then began to engage the scientific community in neurodegenerative disease. And initially, we're thinking about an Alzheimer's disease study. However, the community strongly encouraged them to initially test their hypothesis in amyotrophic lateral sclerosis, or ALS. There were about 30,000 adults in the US and 3,000 in Canada living with ALS. Unlike some of the neurodegenerative diseases where there are decades of progression, ALS is really a disease of moments, like an aggressive cancer, leaving those diagnosed with 2 to 5 years to live. And they spend about half that time actually getting a diagnosis. Currently, there are only 3 approved treatments for ALS, and there are no established diagnostic or prognostic biomarkers in the US and Canada. And finally, from an unmet need perspective, ALS is devastating, universally fatal, and the impact to the person living with the disease and their family is really profound. And so Amylyx undertook a smaller study of 137 participants to compare placebo, allowing people to be on standard of care, compared to Amex 35. And it was a 6-month randomized placebo-controlled period with an open label extension to follow. And the end results of the trial produced a robust data package showing that Amex 35 slowed progression, having a statistically significant benefit on function, as well as a benefit in overall survival analysis in a long-term post-hoc of the extension, including the extension phase.
Michelle, many thanks for outlining the background to AMX 0035. If we can now pass over to Lewis Warrington, the Chief Medical Officer at Ironshore Pharmaceutics. Lewis, could you tell us a little bit about Journée and why and what and where?
Sure. And thank you, Hugh, and thanks to the foundation for inviting me today. And Ironshore's candidate product is Journée PM, and it is the other name is DR-ER methylphenidate, or a delayed release, extended release methylphenidate. So this is a product used to treat ADHD, and as we all know, ADHD has evolved quite a bit in its in the understanding of the disease state itself. The thinking that went into the approach to ADHD from Ironshore's perspective and was particularly associated with duration. The idea was that the evolution of ADHD treatment has been primarily from children, and those children mostly being treated only during school. So that would be during the hours of, say, 8 to 3, and no other coverage. And of course, ADHD is a 24-hour disease state. And so the idea was to try and get as much coverage as you could get, starting in the early morning and lasting all the way through the afternoon and evening. And the approach to that was to create a product that would be given in the evening, mostly around 8 p.m., and travel through the GI tract through the night and arrive at the ascending colon early in the morning. And this would provide coverage for ADHD by methylphenidate upon awakening. And the reason for that emphasis on the morning and making sure that the morning is covered all day duration, including afternoon and evening, is that the morning and the afternoon and evening are a source of distress for children related to school and for adults related to work. And the mornings and afternoons and evenings are particularly compounded by the fact that we have to keep in mind that ADHD is a genetic condition. And so some of the children that are trying to deal with ADHD in the mornings, afternoons, and evenings have at least one parent that is also associated or also has ADHD and may be trying to deal with those issues. Surveys consistently show that early morning, late afternoon, evening are the most severe, and that they rate existing treatments lowest for duration of stimulant effect. And there's a lot of attempts to try and increase duration, for instance, in the afternoon and evening with immediate release. And you have problems with compliance, you have problems with tolerance of that IR, and then also of diversion. And because this is a Schedule II product, you want to make sure that you're not unduly diverting the drug, obviously. A lot of the other formulations, other than that came before Jornay, use differing ratios of immediate release and extended release components. And the immediate release mainly was used because the patients are treated as soon as they wake up in the morning. You need coverage to start, but then there's a lag that's associated with that. The other formulations are absorbed in the small bowel. We're absorbed in the colon, and that makes for a different pharmacokinetic curve and a different ability to gain duration based on dose. As I mentioned, Jornay is absorbed in the colon. It does not contain an IR component, and this is basically because it doesn't need one. You awaken medicated in the morning, and you get a smooth pharmacokinetic profile that doesn't have the peaks and valleys that some of the others do that might lead to adverse events and appetite suppression, that sort of thing. So that was the sort of basis upon which Jornay evolved in its founding.
Okay, Lewis, many thanks for outlining the background to Jornay and where and how it works. Both fascinating stories from Amylyx and from Ironshore. Before I go on to ask Marshall and Lewis a little bit more about how they as companies have attempted to deal with the challenges in the CNS world, can I remind those of you that are dialed into this webinar that we will move on to a Q&A session next. And please, if there are questions, thoughts, comments, either general or more specific to the speakers, please put them in the Q&A box so that we can then moderate those. Marshall, can I come to you first? You're working with a biologic in a neurodegenerative disease. There are certainly challenges there. How would you like to outline that Amylyx has come up with challenges in that space, and what do you think you've done differently in order to get yourselves to market in this clever way?
Yeah, thank you, Hugh. I think the first challenge to recognize in ALS is the history of ALS innovation and how slow it has traditionally been. ALS is riddled with failures, with decades of clinical trials not reaching statistical significance on their primary endpoints. So that was a backdrop of Amylyx coming into the ALS world and designing a study However, lately, ALS has gained a lot of attention, and that's really thanks to the global scientific community, industry collaborations, and the very hard work of the ALS advocacy community to come together to try to identify what the challenges were in preventing success in ALS clinical trials. And this new wave of innovation that's really belongs credit to this increased awareness has provided the availability of new treatments, but also new research and new technologies. We really feel a strong desire to continue to spur along this momentum. Amylyx was fortunate to work with Dr. Merit Sokovich at Massachusetts General Hospital in the Healy ALS Center, and Dr. Sabrina Paganoni, who's also a part of the Healy Center. She was the primary investigator for the the initial study called Centaur, where AMX35 was assessed initially. And we really benefited from looking at the history of different clinical trials and designed a study that using some of the tools that were available. And one research tool that's available is the PROACT database that's also maintained at MGH's Neurobank. And we were able to look at donated samples from previous clinical trials, both placebo and active treated. And we looked specifically at the placebo arm within PROACT. And I used it to help identify a population that we felt in a 6-month period, we would be able to see a difference between placebo and active treatment. And the 6-month period was really important because we'd heard a lot of feedback from people living with ALS and the advocacy community that year-long placebo treatment for ALS patients, it was not acceptable to them. So we really wanted to design a trial where we could find success in 6 months. In addition, their feedback to us was a 1-to-1 ratio of placebo to active is not acceptable because we really want to have as much possibility to have a benefit of treatment, active treatment as possible. So we actually used a 2-to-1 ratio in the Centaur trial. In addition, their feedback was to us, once the trial is completed, please let everyone have access to an active treatment. So we initiated an open label extension period. And post the 6 months, we followed people for an average of 35 months out using the open label extension as well. So at the time, now, today, these are commonly used practices in ALS clinical trials. At the time, when we started the trial in 2017, these were novel. And so we really benefited from being kind of at the right place at the right time, where the experts and the advocacy community were coming together to define what could help create a positive clinical trial. And we were very fortunate that we were there and we listened and were able to incorporate that and continue to receive that feedback. So I think those are some of the things that we overcame. Thinking about the long history, we really just listened to experts and to people living with the disease and advocates to try to design a trial that would be successful. And we were very fortunate that the Centaur trial met its primary endpoint of slowing function on the ALS FRSR.
So that's a really nice bit of a story there, and there are 1 or 2 points I'll come back to. But first, if I could give Lewis the opportunity to see if there are any particular ways that you've come and risen to the challenge of trying to deal with the difficult ADHD patient and carer group.
Yeah, I liked what Michelle said about listening to the patient, and that's what I alluded to earlier, is that coverage for ADHD is incredibly important, meaning coverage during the entire day. It's not this sort of piecemeal approach to just treating children during school and that sort of thing was a huge unmet need for the average ADHD patient. So our approach to that was, in essence, listening to the need that was going on, and that was the need for duration. And by targeting the colon, we were able to leverage the relative inefficiency of absorption in the colon to come up with a pharmacokinetic curve that's entirely different from what was currently seen. As I mentioned earlier, we don't have an immediate release component in our formulation because we don't need one. And that allows us to have a smoother curve that extends farther into the day, and actually such that when you increase the dose, you actually can increase the duration that you can get. And that was a wonderful finding for the ADHD patient to be able to utilize that ability, the prescriber to use that ability to be able to titrate the duration as needed. So the average 6-year-old is going to have a much different need when it comes to duration than someone in college or an adult. So, but because of this sort of unique pharmacokinetic curve leading to a different pattern of coverage, we did need to choose scales and choose the way that we monitored this drug, the efficacy and tolerability of this drug in a different way. A number of previous medications, extended release methylphenidates, have taken one scale and tried to sort of make that fit to each day. And we all know, I think we all know this, think about it for your own life. Your morning is different from the middle of the day, and your afternoon, evening is different as well. So what Ironshore did is patterned the scales that we used to look at the drug in a different way. We used scales that were validated for the morning and validated for the afternoon and evening. So that's thus what we call the bookends of the day, so that we could look specifically at what Jornay was doing for patients during those times. The phase 3 data that we did, we had 2 phase 3 trials. The data were robust. I'll just mention from our Childress study, which is study 1 in our label, response and remission rates were in the 84 to 98% range. And in addition, something that I was very excited about when I joined Ironshore is I noticed that they, in addition to having scales, they also did normative data for those scales. And how many times have we all used a scale and say, oh, there's a 12-point reduction. What does it mean? Here we looked at what does it mean by looking at normative data and comparing ADHD patients to normative patients or non-ADHD patients, so that we could see the ratings that we got. And many times that response and remission that I talked about showed patients were in the minimal impact area of the normative data. So the other thing I'll mention that's kind of interesting, and it's just sort of looking forward, is we hear from the field and from our the people that we work with who are prominent in ADHD, that there are some interesting, I'll call anecdotal aspects of Jornay treatment. And I'll just mention 2 of them. For instance, because of the pharmacokinetic curve that is smooth, as I mentioned before, we appear to see and we hear about less rebound phenomena. So sometimes when patients being treated with a standard stimulant and that stimulant wears off, they might have a rebound phenomenon, and we don't appear to have as much of that. And I'm not trying to say we have none. We're still working on trying to flesh that out, but it appears as though we have less of that, and it would make sense when you look at the way our curve progresses during the day. The other area I'll mention is that we started hearing that one of the main things in stimulant treatment, especially in children, is appetite suppression. Perhaps because of our curve, we theorize, we may have less appetite suppression, and we did hear, in fact, from many people that there seems to be less. And so we embarked on a real-world evidence study to try and look at that very thing, in which we showed an improved weight trajectory in 6 to 12-year-olds versus 6 to 17-year-olds versus Vyvanse, which is an amphetamine. We're working to extend those results currently. So there's a lot that we have learned both in our clinical trial work, as well as some of the things we're hearing anecdotally and are following up on now.
Lewis, many thanks for taking some time leading us through those questions that you resolved. It does seem to be a key learning that both of you, in slightly different ways, have really tried to put the patient and the ecosystem around the patient at the center of your thinking about how you've moved things forward. That clearly, I think, is something that a lot of us are beginning to learn in the CNS space, is that we shouldn't assume what the patient really, really wants rather than what the clinician does. Michelle, do you want to come back and see if there are other comments you could make? I was particularly interested in the way you were saying you'd taken some care to stratify the population on basis of reportage from the community. Perhaps might be an area.
Sure. I think the ALS space and the experts in ALS have really done a really nice job from the leading scientists in the field to capture a number of databases that one in the NCALS database can actually predict survival, and looking at that sort of prediction. The PROACT database that I mentioned, has a number of over 11,000 data points in it from clinical trials that have been completed in ALS and then donated their data to this neurobank. And I would say one of the things that we did in designing the trial was to look at a patient population with definite ALS who were within 18 months of their symptom onset, their first notice of symptom onset. And those two factors allowed us to recruit a relatively fast progressing population, so that over the first 6 months, we could be expected to see a slowing in progression and tell a difference between an active group versus a placebo group. We also expected not very many deaths in that time period in that particular population. As I said before, we allowed individuals who completed the active treatment to then enroll in an open label extension, where we continued to follow their functional as well as overall survival. It allowed us then to have a longitudinal look at survival, simply because we were not able to look at that in the first 6 months. So at the end of the day, there was a statistically significant benefit on function as measured by the ALS-FRS rating scale, but also we were able to do a longer term evaluation and a post hoc analysis showing a benefit in overall survival between the two groups. One of the questions, of course, that comes about is, it's a very small number of people that are in the trial. How do you know that this is representative, et cetera? So we looked at that same PROACT database and propensity score matched individuals in the Centaur trial to individuals in PROACT placebo arm. And we actually were able to very well match the behavior of the ALS-FRS and the loss per month in that scale, because ALS is a relentlessly progressive, nearly monthly loss in function as measured by the scale. And it was interesting because we got nearly an identical rate of loss or rate of decline in the ALS-FRS functional rating scale between the Centaur population versus that PROACT database. So that gave us a great deal of confidence that we were not seeing a fluke in the data in terms of it being a spurious result, because it was very much backed up by the propensity match score in PROACT. We also were able to use that same PROACT database as a placebo control to look at the overall survival analysis. And we were able to use that with regulatory interactions to give even more confirmation of the survival benefit for AMX35 in the Centaur trial.
Just to remind folk who are dialing into the webinar, it'd be great to hear questions coming in to either Marshall or to Lewis, if you'd like to answer them, pose them in the chat box. Marshall, did you therefore feel that the endpoints and the ability to measure that ALS community when you started was as defined as you needed it, or I think I'm getting a feeling that you sort of pulled various different components together to get a better metric than perhaps has been done in the past. So are the endpoints needing to be refined further to make things more robust?
There's a great deal of discussion ongoing in the ALS community today about the use of the ALS-FRS functional rating scale. It has been revised previously to incorporate some other measures. It's been used for decades, and it is clinically validated. It's primarily used in clinical trials. It's not used as much in clinical practice. However, the centers of excellence for ALS do continue to use it. So I think there is a lot of question about it. It is the scale that we have. The emphasis and awareness on ALS in recent years, there was the Act for ALS, which was signed into law in 2021. And there are a number of ongoing initiatives based on funding that resulted from the passage of that act, where people are looking very closely at other rating scales that might be used for ALS. But the ALS-FRS-R is still the gold standard. I think what we were able to do is to use for the first time an external control in the form of the PROACT database to support and confirm the finding that we had, especially relative to the survival outcome with the Centaur results. There is definitely a great deal of talk going on. We also are continuing to study AMX35 and ALS. We currently have an ongoing phase 3 trial called Phoenix being run primarily in Europe. It's over 600 participants using the ALS-FRS-R adjusted for mortality as the primary endpoint, but also including a number of other measures, including quality of life, which is a very important consideration in ALS, EQ5D, some of the staging endpoints, and then, of course, biomarkers included as well, which is another, I think we'll probably get to this in our conversation around drug development in neurosciences, but we're looking forward to the readout of that, the top line readout in 2024 for Phoenix. I just want to make sure that I emphasize that the approvals that we have in Canada, it's a approval with conditions, and that's waiting on the Phoenix data. We do have a full approval in the US. So the Phoenix data is not required for approval in the US based on the Centaur data, which showed a statistically significant benefit in function and also an overall survival benefit, which is the first time that that's been shown in an ALS trial. So we're very much looking forward to the Phoenix trial outcome next year.
Thanks, Michelle. Lewis, and maybe I can come back to you. I think you were implying that you actually did develop some new scales in order to facilitate how you moved Jornay forward. In either a specific or a more general sense, do you think the way we measure and judge our patients and the effect of our molecules is something that we need to look at as a barrier, or do you feel that's something that doesn't require a great deal of change at the moment?
No, I think it needs to be constantly progressing because of the nature of CNS work, and particularly in the psychiatric space, because you're constantly trying to improve how one determines what the actual state is with the patient. And so it can be difficult, especially if you're talking about, subjective scenarios and ADHD, depression, and bipolar, and other areas like that. So I think that can be certainly be expounded on. We had scales in the trials that I mentioned, which were novel in the way we placed them. In other words, other agents were not looking particularly at the morning or particularly at the afternoon and evening, and we did that. So that I think that was a very smart move by Ironshore. It's not without its questions, though, because people generally want to know, how long does your drug last? They'll ask you that, and they usually want a number with hours after it. It's really not about necessarily just the time it lasts. It needs to be titrated to the patient and what their time is. As I mentioned earlier, the needs for a 6-year-old are very different from an adult. The other thing I'll mention too is in terms of trials that are done or required for certain disease states, such as ADHD, the classroom study. If you've never seen a classroom study, they're generally run from 8 a.m. to 8 p.m. That doesn't sound like any classroom that I know of. It's a very long kind of a scenario, and ironically, when you have a novel agent like we do, so our patients wake up medicated. Usually, a classroom study is done that you show up and you get your medicine, and then you start the trial. Our patients showed up medicated, and so we had a little difficulty with establishing a baseline. So, in other words, trying to fit the standard trial designs into every scenario is not always going to work.
Okay, that's great. We've got about 25 minutes left in the webinar, so I'm wondering whether maybe just for the next 10-15 minutes before I come and ask you for maybe some closing comments, whether we maybe just move a little bit more general, because we're all three of us are being working in the neuroscience drug discovery space for quite some while, and I've had, I imagine, more failures than successes in our careers. It's just the way things go. Maybe there's some general comments about how you think we should be thinking about drug discovery. How do we innovate? How do we bring new concepts to the patient to deal with the unmet need that clearly both of you are dealing with, both in the neurological space and also in the mental health space? Michelle, could I come to you first and see if you got any general thoughts?
Sure. I think, we're applying the learnings that we've been able to gather over the last 10 years in ALS, especially, and just thinking about neurodegenerative diseases. Our mission is to one day end the suffering caused by neurodegenerative diseases, and that's a tall order because these diseases are very complex. ALS, for example, is an incredibly complex disease, believed to be probably several disease states within a very heterogeneous spectrum. And the experts in the field, they could talk to you in a much better way than I could about that. But it because it is so complex, there's the belief that it's not just one agent or even two agents that may be able to address all of the different pathologies and pathways that need to be addressed. And in this way, I think if you look at oncology or HIV, there are examples of other areas that have been conquered or have made more progress because they've used a combination therapy approach. And frankly, in ALS, we're really going along that path very quickly. With more and more treatments being approved, there are many more therapies being in research now, with adding on troriluzole and edaravone in our trials, and now Relyvrio is being approved in the U.S. and Albrioza in Canada. When trials are run there, they're typically background. So we are starting to gain that multi-pathway experience. I think another area of innovation, particularly for neurodegenerative diseases, is in really understanding the genomics behind ALS, Parkinson's disease, Alzheimer's disease, et cetera, and bringing some of the technology like antisense oligonucleotides and gene therapy to these areas. We actually are embarking on that as well, going beyond AMX35. We're investigating a compound we've named AMX114 that is an internally developed ASO that targets calpain-2, which is a critical effector of axonal degeneration in ALS, which is a downstream from where we're studying in the ER and mitochondria. So that's an example of thinking about divergent targets, and it's not just going to be a one drug cures everything. So I think that is a particular challenge in neurodegeneration right now, and probably in other areas of neuroscience as well.
That's some very nice observations in there. I certainly like what you were saying, was that even within your neurological space, where maybe you've got one diagnosis, you believe there may be several biotypes that are operating in that space, and you're going to need different pharmacologies to address that. Lewis, in the mental health space, ADHD, attention hyperactive disorder, none of that is biologic. You actually made your comment yourself that actually understanding how the treatments work may actually tell us something about how the patient's biology is. Would you agree that maybe we need to bring more biology into the mental health space rather than just purely describing the patient symptomatically?
Well, in my career, that has been occurring, over time. I mean, just some of the comments I made earlier about the genetic aspects of ADHD. So there is a final common pathway of some kind. We just haven't found it. We know it involves dopamine, but not sure. And we know treatments act on dopamine. So we know that there are going to probably be elucidations in the future on different aspects and getting to etiology. And I think the treatments do help to do that, and to think of in other psychiatric fields, major depression, and bipolar. I mean, there have been a number of therapeutics that have come out that have sort of shed light on that as well. The other thing I want to mention was that I think as time goes on, we need to involve more, and I'm thinking about ADHD when I say this, but I think it also applies to your area, Michelle, is the family support and the families that are involved. So certainly for the field of ADHD, you have a lot of family involvement, that where the treatment that you bring to the fore and how that manifests, certainly impacts the family. We've done surveys within our shore where we've seen the impact on families that subpar treatment can give to a family, and we actually included a questionnaire in one of our studies on the caregiver to sort of see the manifestation of good treatment on that caregiver, and this is done in other fields like Alzheimer's as well. I think we need to look at that aspect of it. And then what I'd like to see in the field of psychiatry particularly is more health outcomes and research on what does a good outcome look like. I mean, we've been talking about it here, but how much positive outcome can we expect in ADHD, for instance, just to use that? Is it based on functioning? Certainly is, but what functioning and what kinds of skills do we need to bring forward to look at those? So I think it's the sort of old days of thinking about just sort of symptomatic and can I dampen that symptom and get a good score on scale. It needs to be broadened, I think, to encompass a lot of different things that have to do with that particular patient.
No, I couldn't agree more. Certainly, if we're going to innovate, we need to bring new mechanisms and approaches, but also we've got to show real-world benefit rather than just a change on a clinical scale. That's something I think has come out from both of the stories you're telling, is that getting the patient and the carers around that patient to agree that something real has happened that is vital. I liked your story, Lewis, about the school test not running from 8 a.m. to 8 p.m. That's an abstract formulation. So, Michelle, do you feel we know enough biologically about ALS to stratify, or do you think actually by being able to use AMX that you may actually be able to begin to trigger new research to maybe find ways of biologically stratifying those populations so that your next round of molecules will be more precise, more effective, more rapid?
We're just in the beginning stages, especially with AMX 35, of like Lewis said, starting to get broad exposure. The benefit of having a conditional approval in Canada and an full approval in the US is, several thousand people since our launch have now been placed on treatment. And that means that they're out in the real world taking the product, and it's also expanded to a broader population rather than the narrower populations that we study in clinical trials. So we will be undertaking an aggressive and broad real-world evidence strategy and approach. And we're luckily, like I said, there are several within the United States and Canada, several databases where we can go and find records of people taking MX35 and look at various, it depends on what's recorded in the database, but there are some ways we can also prospectively identify. From the mechanism of action of MX35, there's no reason to think that any person living with ALS, no matter their stratification from a biology perspective, might be able to benefit. But we haven't studied that, so we don't know it. There has been tremendous progress in stratifying from a biological perspective what the different various phenotypes of ALS might be. I think the funding and the attention and awareness for doing that has now, there's a period of time when the Act for ALS in the US is going to have a very focused amount of money spent on that through the NIH, for example. There have been several grants funded recently for very large multi-center, some of the world's leading experts in ALS to take a look at some of these questions. And then even from a basic science perspective as well. So I think we're at the cusp of really getting that research started and bringing together the world's experts and giving them the resources that they need to answer many of these questions. So I think, if we spoke several years from now, hopefully we would be able to describe, in more detail what that might look like. But I think from an MX35 perspective, I'm not sure it's a strong tool. I think what we can say is that we can look to see outside of the populations we've studied. For example, there's a lot of focus and rightly so, on the 10% of ALS cases which are of genetic origin. 90% are sporadic. They have gene mutations that are common across even within the sporadic, but there are targeted treatments for those genetic causes. We need to answer the question, does AMX35 also work there? And there's no reason to think that targeting the endoplasmic reticulum and the mitochondria added to a genetic treatment would not also be beneficial, but we can't answer that question until we look at the data. And probably our best way to do that, because these cases are so rare from a genetic perspective, is to look at real world evidence. So I think we can answer some of those bigger questions by looking at that data as it evolves, because it's going to grow longitudinally.
Very nice, thank you. Lewis, historically in mental health, we've often developed drugs which have kinetics, and our clinical colleagues try and persuade us to get 90% 24 hours a day, 7 days a week, because everybody assumes more is better. You've been very clever to use the physiological system and the circadian cycle to optimize the way that your molecule is working for patients' benefit. Do you think that just looking at pharmacokinetics gives us an opportunity to reinvent certain areas of mental health, so we actually try and understand the patient even at that simple level better?
I would go so far as to say we have an obligation to do that, to look at the pharmacokinetics as one factor, but there are others. I liked your point earlier about etiology, but there are things that you can do short of etiology. We all want there to be a known etiology that we can target that. But in the meantime, what does one do? You can search for greater efficacy, greater coverage, as I've mentioned for Jornay, but you can also look to try and make things better for the patient from the perspective of side effects. And one of the things that I remember from my fellowship, my child and adolescent fellowship, was everyone who came in on a stimulant, and stimulants work very, very well, but they have a downside in that they cause adverse effects such as insomnia and appetite suppression. And one of the things that I think we've been able to do with Jornay is advance the idea of being able to give targeted therapy, and we certainly have the data to show that, but also to dodge some of the common, or try to dodge some of the common adverse events, such as I mentioned earlier, appetite suppression, because it's a really big one, and especially for younger children, because you don't want to dampen a growth curve and not be able to get that back. So I think just to broaden that concept into sort of the psychiatric space at large, I think the idea is to certainly try to get to etiology, but also in the meantime try to find ways to get the disease improvement that we want in the disease state, and try to dodge as many adverse effects as we can. That's why I always have difficulty with the concept of the Me Too. I was called by a colleague from Europe, and they have very stringent ideas in Europe about, you must be able to show superiority over the common, over the standard treatment in Europe. And I was told by this individual, they said, why are you over there with so many different agents on the market? And I said, well, they're not Me Too's, they're showing improvement in certain areas. And so even if that's adverse events, if you can make a drug better, more tolerable, that's an advancement. So I agree totally with what you're saying, that we need to search for etiology, but in the meantime, we can maybe make things easier for the patient by making their agents more tolerable.
Very laudable end, and I entirely agree with you that engineering it so that it's ideal for the patient. Maybe even, I think you're doing yourself an injustice by saying you're dodging the side effect. I think you're actually designing it to avoid, which is perhaps, we ought to give you the credit where credit's due in that regard. We're moving into the last 5 minutes of this webinar. what I'd like to do is just thoughts relating to either AMX 0035 or to Journey to the table. And maybe Marshall, can I ask you to go first and see if any last comments you wanted to make on behalf of Amylyx?
Sure. I again just reiterate on behalf of Amylyx our thanks to have the opportunity to join you on this webinar. I really enjoy this dialogue, and it really fosters thinking about how we can think about future drug development, and particularly for us, we think about neurodegenerative diseases. Obviously, we are committed to working with the ALS community, regulators, clinicians, people living with ALS, and the really active advocacy landscape. And I think you prompted me to think about, to learn about ALS using MX35, but we also are looking at other neurodegenerative diseases. We're currently have announced that we're going to start a trial in progressive supranuclear palsy, PSP. It's interesting. There's a compelling scientific rationale, as there's overlap across many neurodegenerative diseases with ER stress and mitochondrial dysfunction. Also, there's biomarker evidence of MX35 having a reducing effect on tau, which is the hallmark PSP protein. In addition, we're looking at Wolfram syndrome, which is a very rare recessive inherited disease that results in neurodegeneration as well. So we're hoping that as we branch out and explore other neurodegenerative diseases, we learn more and more, and that we can apply it broadly, but also specifically to ALS, as we're really committed to continuing to work there, and hopefully one day have combinations of therapies that can help people living with ALS live much longer and higher quality lives.
That's super, Marshall. Lewis, can I encourage you to see if you'd like to make some comments from Ironshore?
Yeah, so on behalf of my colleagues at Ironshore, thank you for the ability to come and speak today. I would say that one of the passions at Ironshore is the patient, keeping the patient focused, and we feel that we've done a very good job of that with Journey, but that's not where we don't stop there. We want to take the delivery platform that we have and advance that potentially into other areas, with a focus on the pharmacokinetics and delivery that would afford other disease states either a better, sort of getting at the etiology, or even a better sort of tolerability platform and timing of drug delivery. So we're continuing on that quest. And again, thank you very much for allowing us to come on and speak today.
Well, I'd like to thank both of you for telling us so much about the exciting science you're doing for your two companies, and showing how by innovating in the appropriate spaces in the appropriate ways, we can provide better outcomes for the patients in whatever area it is that we try to direct our approaches, both in research and discovery. And then on to development. Clearly, in the CNS space, we have challenges, but both Amylyx and Ironshore have risen to those challenges, and you successfully progressed through those. And I very much appreciate your comments to the wider questions that hopefully will give the whole community the ability to move forward. Just to close, can I remind remind everybody that's dialed in that on Thursday, on the 26th, the Galien forum will actually be completed. So the end of this exciting week of webinars and presentations will come to a conclusion. There will be some excellent talks, and actually one will be actually on the mental health state in America, which sort of comes into that neuroscience space. And then the really exciting bit when the prize awards will be announced. So I encourage everybody to dial in for that and find out how things have progressed and who've been the very meritorious winners this year. So thank you very much, Marshall. Thank you very much, Lewis. Thanks to you that have been listening, and I wish you all the very best wherever you are in the world, and hope that we can bring new drugs to patients at the right time for the right reasons. Thank you.
Thank you very much.
Thank you.